This is our presentation over Chapter 8 in your textbook. It covers fungi. So let's take a look at the fungi. Um, Looking ahead in this chapter, what you should expect to learn is to appreciate some of the novel features of fungi that encourage scientists to place them in their own kingdom. You should understand the sexual and asexual reproductive cycles of fungi and compare those cycles to those of other microbes. You should be able to explain how fungi benefit society in the production of various foods, the synthesis of key industrial products, and the cycling of elements in the environment. You should be able to describe several important fungal diseases that affect plants and humans. You should understand the nature of yeasts and the biochemistry of their metabolism as it relates to bread and the fermentation industries. You should name and describe 4 groups of fungi and identify key members of each group. You should recognize the important roles assumed by fungi in lichens and in mycorrhiza. Talking about fungi in general, um, fungi may have filled a niche during the dinosaur extinction because not only did dinosaurs die out, but plants died out as well. And fungi have a great mode of survival. They are saprophytes, so they can feed off of decaying animal and plant matter. And in an environment where practically everything has died, this is beneficial. We can look at some images below, and you can see a view of a yeast, which is a eukaryotic cell, 2 molds that are wrapped together, and kind of the parasitic relationship where a mold will grow on a fruit. Now, molds do like living off of dead organisms, and they also like living in acidic environments, which we will address just a little later on. In general, the need for fungi. They are ecologically important as decomposers. Many of them are saprophytes, and a saprophyte is something that lives off of dead organic matter, and as a result, can recycle it back into the environment. They break down complex organic molecules into simple molecules that get to be used by other organisms, and as a result, they keep that carbon cycle and nitrogen cycle functioning. Now, some classifications of structures and growth of fungi, there's a few terms to know. First off, yeasts, and we'll talk about them a little bit later on, but these are fermenting type organisms that can produce carbon dioxide, alcohol, byproducts like that. Molds, we're familiar with molds. They tend to grow in kind of this moist environment. We have hypha, which are strands of cells of any kind of fungi. And a mycelium is actually a mass of those strands. Now, the study of fungi is known as mycology, and a disease caused by fungi is mycosis. So these are terms to keep in mind as we progress through this chapter. These are eukaryotic organisms. They contain in their cells nuclei and other organelles which allow them to survive. Many of the fungi form spores, in fact most of them. They don't contain chlorophyll. They produce sexually and asexually depending on environmental conditions and other factors. The cell walls contain chitin, which is a complex carbohydrate that we see in animal life, such as crabs and other shellfish. And between the cells, in the hypha, so we've got this string of cells, we have septa, which are incomplete walls between cells. So we have a sharing of cytoplasm, sometimes cytoplasmic organelles, and other substances between one cell and the next. We mentioned that many of the fungi are saprophytic, that is, they live off of dead matter. So that also means they're heterotrophic. They have to eat something. Now, the fungi have digestive enzymes, secreted outside of the cell, so they don't gobble up something through phagocytosis or anything like that. What they do is they squirt material on the surface and let it dissolve, and then they can absorb that. So simple breakdowns products are absorbed by the cell, which the cell can then use. Among the enzymes that are used by fungi are cellulase, and cellulase breaks down cellulose. Cellulose is a very hard material to break down. That's the stuff that plant life and trees are made of. Cattle have the extra stomach, the rumen, full of bacteria that can break down cellulose by using this enzyme. Termites have this enzyme, but there's not that much that can really break this down. So fungi are very beneficial in returning stuff to the environment. They do something kind of different from plants. They store glucose as opposed to starches. Plants store starch as an energy source. Animals store glucose, and they store theirs in the form of glycogen, which is exactly what we store in our livers. As I mentioned before, most fungi do exist as saprophytes or saprobes, so they live off dead material. But there are some fungi that exist as parasites, and we'll look at the parasitic relationships and how that affects us a little later on. So generally, we said nutrition of fungi. Yes, a lot of them are saprobes. So fungi tend to like to live in acidic conditions. That green stuff that's on the orange in your refrigerator that's been left there too long, that's a fungi. And it likes that because it is acidic. So fungi grow in some familiar acidic foods. We see them in sour cream and yogurts and citrus fruits, most vegetables, breads, and cheeses. We see these kind of molds and other things form. Now, sometimes that's good. There are favorable contaminations because blue cheese uses Penicillium roqueforti to make the special characteristics of blue cheese. Other things that we do, the carbon dioxide that we put in colas comes from fungi. We brew and ferment sake with fungi. We have fungi in blue cheese to give it that unique characteristic. Fungi in the form of yeast are used in bread production. So fungi are very important both environmentally and in industrial uses. Now, we've categorized other organisms based on the way they moved. So motility, we've based categorization on shape and cell membrane type. In this case, we categorize fungi based on their type of reproduction. So there are asexual reproduction cycles and sexual reproduction cycles. We have a few different groups. Actually, I'm going to skip forward here, and we have the zygomycota, the ascomycota, the basidiomycota, and the deuteromycota. I'm going to jump back and just talk about what this means. See, in asexual reproduction, there's a couple of ways that can occur. The plant can form something called arthrospores, and what happens there is that the hypha, the parts of these strands fragment, and each fragment or cell that has fragmented off of that becomes a new hypha and eventually a new mycelium or stack of microorganisms. And then we have a different process where we have chlamydia spores. Spores from chlamydophores are in a sac structure called a sporangium, or they can be located on the tip of a specialized hypha called a conidiophore. And if that's the case, then the spores are called conidia. Now, asexual production has the advantage of giving a large number of spores, and they can rapidly spread just because of the numbers involved. Unfortunately, there's not a lot of variation genetically, so if the environment has if they've had to go into a spore mode because of environmental changes that are widespread, this doesn't necessarily ensure survival. So we've talked about the basic modes of asexual reproduction, but there's also sexual reproduction. They have a fully functional genetic makeup, but our cells are diploid, as they have two copies of genes. In fungi cells, they have one copy, but there are phases where two cells will merge together and you will get this diploid cell. So what happens is cell fusion, spore formation, and you can see this in figure 8.5 of the text. But the cells of fungi, which are normally haploid, that is one set of chromosomes, the haploid parent cells fuse together to give a diploid progeny called a zygote. And that's the same term that we use for that single cell that has both parents' genetic makeup. Now, unlike our cells, which undergo mitosis, the diploid cell or zygote undergoes meiosis, which creates haploid spores. And in that process, the genes from each parent are mixed. So each child spore gets a unique set of genetic material. It has all of the genes necessary for its function, but some of those genes came from, say, one spore and one comes from the other, or from one of the parent cells and one comes from the other parent cell. The spores that are made are environmentally resistant. They can survive through bad weather and things like that, just like bacterial spores. But when the conditions arise, the spores are able to germinate and create a new haploid organism. Now, this process results in fewer spores, but they are more genetically diverse, and so there is a greater chance that one of those spores will survive if there have been some environmental changes. So, basically, we mention this because we classify fungi based on the sexual reproduction techniques that they have. Just as we've classified bacteria based on shape, we've classified the protozoans based on the way that they move. We classify fungi based on their reproductive cycles. In asexual reproduction, we end up with spores. And these spores can be formed on a couple of structures, either a sporangium or a conidiophore. And we end up, in the case of a conidiophore, with a structure called a conidium. These are environmentally stable, like bacterial spores. They produce extremely large numbers of spores, but all of the spores are genetically identical to the parent cell. So we get the advantage of numbers, but the lack of diversity. Now, general divisions of fungi. What we have are the zygote fungi, or zygomycota, and these make diploid spores. We have the ascomycota, which develop diploid spores in a spore sac. Basidiomycota, also producing diploid spores. These are what we think of as the mushrooms, and they have spore-producing bodies of the larger fungi. Now, the Deuteromycota, we've been able to identify the asexual method of reproduction, but thus far there's not been a method of sexual reproduction that has been identified, and that's the key difference there. Okay, so we've talked about the various characteristics of the fungi thus far. We're going to talk about the specifics of the 4 types of fungi in our next presentation. Thank you.